supported by this project. So when we have a look at our center, we do have more than 50 academicians coming together under the center, and it is established in 2009. And this is an important vision academically, of course. And we do have an important vision. So in some certain areas of life sciences, we would like to come up with some specific, unique studies. And for that reason, we are providing very comprehensive projects, publications. We do have this experience under this center. So this makes our foundation strong. And at the moment, starting with cancer targeted treatment technologies. So this is our umbrella theme. So it is, we are trying to become a leading research center in the world. And there are smart medication transportation systems, vaccine or microsystem-based smart devices, molecular imaging methods. These are the research projects that we are executing, which can be leading and exemplary for our country. And we do have more than 200 PhD and master's degree students, who are supporting our center. They're working with an academic staff. They are also located in the different faculties of the science faculty and in some other departments. And in the life sciences of Turkey, we are working on the future technologies from today. So we're trying to develop infrastructure, and we're trying to keep our works up to date and accelerate all our activities in this center. Of course, we're not just focusing on academic studies. Also, we're focusing on health sector and also health entrepreneurship ecosystem. So we are providing some very important supports to these sectors too. Here you see the management board of our center. I am very thankful to them. They are allocating time for this center, and they are exerting their efforts to keep the center alive. So we do have assistants, experts, secretary, who contributes to our center. So here you see the development of our center in time. So this is a 13-year-old adventure, which we can say. But regarding academic development, as you see, in the suprastructures, we do see some units. So from the beginning, we do plan all of them. And with patience, we have found some extra funds to realize all these. And of course, we are also dealing with some extra projects, certifications and plans. So it is like a real interface for the industry and for the future. We are trying to define the gaps for the future. And also we do have some national and international funds which are being assessed. And life sciences had 3 main areas, which is animal production and care unit, clean room, and test analysis unit. Our animal unit for production center, for production purposes, it is certified, and also we do have the IALAC full accreditation, and since 2017 it is keeping it up to date. So in terms of animal sciences, it is very important. So it can be a small scale lab, but in terms of quality and in terms of its services, it is one of the leading ones in our country. Clean room unit, again, it is in Kandilli campus. There we do have microsystem-based devices. We are developing these devices. So active executor devices are our main theme. There is an important infrastructure here. And MEMS sensors and the other active optical and electronical devices are being integrated to medical devices here. And we haven't neglected certification here. We do have the ISO 13485 certification for this room, and it is being kept up to date. And here, the enterprises and SMEs are being provided some specific clean room unit services for their activities. And this is test analysis unit. This is where we convene different devices together. They are working in coordination here. And of course, we are dealing with physicals and chemicals here, tests, physical, chemical tests and characterization. And from our website, you can receive the detailed information. And under this EU-Turkey cooperation, and under the coordination of the Ministry of Industry and Technology, so there is a competitive sectors program, and we do have an extra support from that program. And we are supporting clean room, vivarium, test analysis units. So we have made some very important additions to these units, and we do have an animal imaging lab, and also we do have a pilot production infrastructure. And also in relation to our center in Sarıtepe campus, there is another center lab for microalgae bioproduction. We are also supporting that. All this center is known as Boğaziçi Life Science Center. So its own ecosystem is distinct from the other centers in Turkey because it is working with a health cluster. So this is Istanbul Health Cluster, and in accordance with their needs, we have designed this project. So the infrastructure, the users of our infrastructures are defined academically and also industrially. So within the existing building, so under Life Science Center, we do also have the health industry clustering, Kandilli Health Accelerator Program. To support the entrepreneurs, we have positioned this platform here. Istanbul Development Agency is supporting this. So we are merging the IPA support and Istanbul Development Agency support together to provide synergy for the members of this sector. And also there is another sister center, which is Technopark Istanbul BioCube. It is being coordinated by another unit of the Ministry of Industry and Technology. And similarly, we are supporting entrepreneurs, and we're trying to fulfill a gap here, especially in Kandilli campus. I have explained to you how this project is positioned. And also there are some pilot production centers in Technopark, how we're integrated to BioCube and BioIncubator. And also we do have the other microalgae project in Sarıtepe campus. And regarding these infrastructures, we do have very serious preparations in summary in Istanbul in the field of health, from academy to the SMEs. So we are acting as a bridge to develop the health centers of SME. So this year will be a breakthrough year to do this. I would like to extend my thanks to the EU, the Ministry of Industry and Technology, and all the contributors. And also to the technical assistance team, of course, first to Pascal and also his team. And this is where I end my presentation. Now I'm going to listen to you. Thank you.
Thank you very much, Professor Cengiz Öztürk, for this great presentation of the center. I will carry on with the presentation on the technical assistance project. We are collecting all the questions on a separate track through the chat of this Zoom platform, and I will come back to all the questions at the end of the webinar. So I will share my screen again. I think this is this. Do you see my screen correctly? I guess, yes.
Yes.
Okay, thank you. So I will carry on now the presentation of, with the presentation of the technical assistance project that is funding this webinar. And afterwards, we will move on to the webinar with the talk from Professor Mangala Srinivas. So let me start with explaining briefly what is this technical assistance project. This project is called Technical Assistance for Research and Development Support Labs for Life Sciences SMEs towards Global Competitiveness. This is a project co-financed by the European Union and the Republic of Turkey under the Competitive Sectors Program executed by the Ministry of Industry and Technology. This project is 18 months long. It has started on the 1st of November 2021, and the base of operation is Istanbul in Turkey. The overall objective of this project is to accelerate the research and the development activities of SMEs active in the life sciences domain, with a focus on specific cutting-edge technologies and high-value-added products. In practice, as you can see, the building of the Life Science Center is depicted in this slide. It was also presented previously by Professor Öztürk. The purpose of this project is to augment the current technical capabilities and to add new infrastructure to the Life Science Center, providing links to the regional industry with a focus to improve its cooperation with the regional industrial partners, more specifically to expand the support services regarding preclinical animal imaging experiments and specific material characterization tests to the local SMEs involved in specific R&D activities related to in vivo medical devices, biotechnological products, and biomaterials. And I also would like to mention the pharmaceutical industry sector. So the parties involved in this specific project are the Ministry of Industry and Technology, with the Directorate of EU Financial Programs, the delegation of the European Union to Turkey. Both of these offices are located in Ankara, in Turkey. The recipient of these services of this project is the Boğaziçi University in Istanbul, and the contractor is System Global. This is the firm that is in charge of implementing this project inside the Boğaziçi University. So in a single slide, I will try to summarize in a single slide the different activities of this project. So as mentioned previously, you can see on the left side of the slide the existing units that are providing services to SMEs and industry. From the center, we have clean rooms, a vivarium, test analysis units. And these 3 units will be complemented at the end of this project with 3 additional units: a small animal imaging unit, a pilot production unit, and microalgae bioproduction unit. In parallel to this expansion of the center, the project supports startups and small SMEs in the pharmaceutical industry, medical devices, and biotechnology by providing them support of consulting, support for business development and expansion of their own businesses, but also training on the different activities of the center. So these activities can be summarized in this slide with 2 sets of components. First, a component A that is depicted in the left side of the slide, which is really the provision of support to the SMEs, with not only an analysis of the SMEs and SME landscape of the region, but also provision of market analysis specifically towards providing opportunities to SMEs through the organization of focus group meetings. And as I previously mentioned, we will provide support to startups and micro SMEs as consulting and training on the different services. A different part of the project is associated with the development of the center itself. And we are currently reviewing the center's overall operations. We are going to establish a business development unit within the center that will pilot the provision of services to the SMEs. And of course, we are running development of communication and visibility activities in order to disseminate all this information to the future users of these services. This project provides a lot of opportunities for the region, for the center, and we have now 18 months to set up all these new services within the center, and it's a real pleasure to be able to provide all these services to the SMEs and to the center. So, with these words, with this final slide, I will close my presentation. I would like to thank all the assistance team of the project. This list is non-exhaustive, and we are involving more and more experts that are coming to the center to provide support during the timeline of the project. I would like to thank the members of System Global who are actively supporting the project and its implementation in the center. And of course, I would like to thank all the members of the center that are helping us and opening their books, basically, so that we can really provide an expansion of the center. With these words, I would like to close my presentation, and I will give the floor to Professor Mangala Srinivas. Mangala has recently started as professor and head of department at Wageningen University and Research in the Cell Biology and Immunology Laboratory department. And Mangala is founder and CSO of Senia Imaging, a spin-off from her academic group in the Netherlands. Mangala started her career in research at the National University of Singapore before completing her PhD at Carnegie Mellon University in the USA. Her work focuses on agents for in vivo imaging. Mangala's early work helped establish the field of F19 MRI for quantitative in vivo cell tracking, publishing the first paper showing that this was possible. More recently, her group works on customizable nanoparticles for imaging and advanced personalized medicine applications, such as cell therapies and immunomodulation. Some of these nanoparticles are produced at GMP grade for clinical cell tracking using multimodal imaging in melanoma patients. Her group works with fluorescence, MRI, PET, SPECT, ultrasound, and photoacoustic imaging in different disease models, particularly cancer and cardiovascular disease. Recently, she won the Dutch Venture Challenge in 2015 for her ideas on cell tracking. Overall, Mangala's team has taken a new type of imaging agent from development to the clinic and commercialization. And so she represents a really model as a scientist, and I'm really interested to hear her talk now. So, Professor Mangala Srinivas, are you ready to share your screen?
Thank you, Pascal, for the really nice introduction, and thank you to all the organizers as well for this invitation. Let me just share my screen. So here we are. Okay. So let me get started, and I hope this talking speed is okay for the interpreter as well. I guess so. All right, good.
Don't worry, go ahead. Thank you. Thank you.
All right. So as Pascal said, I'm now head of the cell biology. Work was done through my spin-off company, Sanya Imaging, or in previous position at the Radboud University Medical Center at the Department of Tumor Immunology, which is also in the Netherlands. So at the Department of Tumor Immunology, we specialized in dendritic cell or DC vaccinations in melanoma patients. And over the last 2 decades, we have vaccinated over 300 patients. What this involves is the isolation of therapeutic cell precursors from the patient that are then activated and matured, antigen loaded, and then injected as therapeutic cells into the patient. This therapy has been successful, and what you're looking at here are some CT scans of patients with liver metastasis, which is highlighted in blue. And you can see after treatment, the metastasis, the tumor burden went down. And also there was more presence of anti-tumor T cells in the patient blood. So the therapy can work. And if you look at this on a larger scale, if you look at overall survival of patients shown in blue who received therapy versus matched controls in yellow, you can see that the treatment does work well in patients who have already failed other therapies. But what I'm interested in is why is it that some patients do better than others when all of them are receiving the exact same therapy? So to answer that, we need to understand what happens to the therapeutic cells in vivo. And this applies not just to dendritic cells, but to all kinds of cell therapies, including Carti cells or stem cells and so on. And this is a multi-scale problem because we need to look at the whole body level, at the level of the cell, and also inside the cell in order, excuse me, in order to understand what is happening with the kinetics of the cell. For example, where are they migrating to? Are they getting to the site of interest, in this case, the tumor or a lymph node? Are they functional? And are they able to do what they should? Are they able to divide and to degrade in a non-toxic way? So to try to understand why cell therapies have a relatively poor success rate, we need to answer some really basic questions in a relatively fast manner. So just simple things like, are the cells in the right location? Are they alive and are they functional? And these are really basic questions for which we need to have answers in a non-invasive manner. So ideally, we would be able to quantify the numbers of cells at the region of interest, such as a tumor or the lymph node, over a relevant period of time, which can be days or weeks. And to do this in a way that is non-invasive, without biopsies or without cutting the patient open or to have invasive samples. There is a lot of need for cell tracking, both academically and in the commercial field, because cell therapies are simply very expensive. Some cell therapies, like CAR-T cell therapies, can cost around $500,000 per patient. And this is just, it's a huge cost, especially if you do not know that it is going to work. So being able to track the therapeutic cells will help us determine their biodistribution and to assess their functionality on a per patient basis if needed. And this is how specifically imaging can help with that. So what you need to do then is to label your cells with some kind of imaging agent. And then the cells are injected into the patient. And then we can look at the pharmacokinetics and biodistribution in a longitudinal manner using non-invasive imaging in order to get information on the in vivo functionality and in order to lead to better outcomes for the patient, but also for the cell therapy developer. In short, imaging provides biomarkers in a non-invasive manner. And the imaging data, it can be non-invasive, it can be quantitative, longitudinal, kinetic. Depending on the imaging modality, it can be in real time. Many imaging modalities are translatable to the clinic, so they can be used both in a preclinical and clinical setting. And they can give us information both at an anatomic level and at a functional level. There are many different parameters that need to be optimized in order to optimize a cell therapy. So, and this depends on the cell therapy developer, whether you need something that can be used in humans, what level of sensitivity you need. Are you able to deal with radioactivity, or is it not suitable in your setting? The availability of imaging modalities, the cost, the resolution that you need, and so on. And concurrently, there are also many different imaging modalities. Pascal mentioned a few of these. There's SPECT, PET, ultrasound, MRI, so on. And the thing to keep in mind is that none of the imaging modalities are perfect. You get different information from different imaging modalities, and being able to pick the imaging modality that you need for the questions that you have is really important. And this may mean that you need to combine different imaging modalities in order to specifically answer your questions. So here's an example of how imaging can help with cell therapies. So this was actually the first paper showing the use of MRI-based cell tracking. This is dendritic cell therapy, and the dendritic cells were labeled with an iron oxide agent, SBIO, and these result in dark spots. So these cells were injected directly into the lymph node of a patient, and that's what you're seeing with the arrows. And we couldn't see that once the cells were injected in the lymph node becomes darker and the cells are present. So that was really successful. But actually the main finding of this study, rather ironically, was that in almost half of the patients, the cells were misinjected. So they were simply injected in the wrong place. And what this means is when you look at the patients, if we had not done imaging, you would only see that the therapy did not work, but there are millions of reasons why a therapy like this might not have worked. Without the imaging, we would not have known that the cells were injected in the wrong place. So this is a very clear example of how imaging can help with cell therapy optimization. Another example is looking at functionality. So this again is dendritic cells injected in the lymph node of the patient, and then we injected a PET tracer FLT, which is a nucleotide analog of fluorinated thymidine that accumulates in dividing cells. So the idea was that if the dendritic cells trigger T cell division in the lymph node, we should see accumulation of the PET tracer. And that's exactly what we saw in the injected lymph nodes. There was an accumulation of the PET tracer, which we did not see in controls that were non-vaccinated or mock vaccinated lymph nodes. So that was a way to look at functionality of the cells, again, using a non-invasive technique. My interest particularly is in quantification of the numbers of therapeutic cells. And we are doing this using magnetic resonance imaging, or MRI, using the 19F or non-radioactive fluorine nucleus. So with MR, we are able to see the anatomic background in grayscale, and using conventional contrast agents like iron oxide or gadolinium, you can see a change in contrast, so in the grayscale of the anatomy. With fluorine, you're looking at essentially a different color. So this allows you to superimpose the fluorine signal on the anatomic background. And this allows for non-ambiguous detection of your labeled cells, but also for direct quantification. And that signal intensity is directly proportional to label concentration, which is not the case for traditional contrast agents like iron oxide or gadolinium. And this allows us then, from the image data, to quantify the numbers of cells. So here's an example. This was the first paper showing fluorine-based quantification of cell numbers in mice. And what you're looking at here is a diabetic mouse that was injected with labeled T cells that home to the pancreas in P, labeled P here. And then we used an external reference for quantification. So from the raw fluorine image, which is the color image that you see, we can then quantify the numbers of cells in the region of interest. And in this case, in the pancreas of the mice. We have also done this on a clinical scanner with human T cells or with human dendritic cells. And in our settings with this cell type, we can detect a minimum of 5,000 cells per unit volume within an imaging time of 10 to 15 minutes. But this really depends on the cell type, your imaging hardware, and how long you can image for. So how are cells labeled for fluorine MRI? We use compounds called perfluorocarbons, which are long polymers of carbon, fluorine, and sometimes with oxygen. And these compounds are very interesting because they are simultaneously hydrophobic and lipophobic. So this makes them quite difficult to work with. Which means that if you mix perfluorocarbons with an organic or an aqueous phase, you get a third phase, which is a fluorous phase. So they need to be stabilized in some manner to make them accessible for in vivo use. At the same time, they have a long history of clinical use as blood substitutes. They are also known to have a very slow clearance in vivo, which can be difficult for translation. Perfluorocarbons have been used mostly as blood substitutes in the 80s and 90s, where they were injected in large volumes intravenously in humans because they dissolve oxygen. And this is a very unique property of perfluorocarbons. They're also very inert, which means in general, they are safe for biological use, but they can have slow clearance times. So the agent that we're working with is a nanoparticle of about 200 to 250 nanometers in diameter, consisting of a polymer, which is an FDA-approved polymer, polylactic glycolic acid, PLGA, and trapping a ring perfluorocarbon that is extremely inert and that is not metabolized in vivo. So here's an example of the production technique. So this is not what you're looking at is not GMP grade, but it's an upscaled technique for continuous production. It was challenging to build this because we have three phases. We have the aqueous, the organic, and the fluorous phase to work with. So it's quite a unique system, but this allows us to produce relatively large amounts of the nanoparticle, both at GMP and non-GMP grade as needed. So the process involved three phases, as you can see, the PFC, the organic phase, and the aqueous phase, which are then mixed together at high energy. So with the basic particle, we can carry out MRI, fluorine MRI. We also see them in ultrasound, and we include an FDA-approved dye, IC green, which allows us to see them in photoacoustics as well. For preclinical use, of course, we have a lot more variability in what we can do. So we can adapt the diameter, we can add different cargo, for example, different drugs, or modify the surface charge and so on. So because we have more space to play in a preclinical setting than in what is approved for clinical trial use. So here's an example of what an MRI scan looks like. So you need a subject, in this case, the mouse is anesthetized so that it does not move during the imaging. It's placed in a holder. We monitor the breath rate and the heart rate. We place an external reference for quantification, and then the mouse is put into the scanner. It's not a painful process in any way, but we need the mouse to be still, and that's why the mouse is anesthetized. And the same mouse can be imaged at multiple time points once they wake up from the anesthetic, which allows you to acquire longitudinal data from the same subject. So here are some examples of data. So this is the images on top are of the mouse heart, and all of these were in the same mouse. This is a mouse that had a myocardial infarct, and in the mouse heart wall, which is outlined in white. And then we injected the nanoparticles intravenously. It's taken up by phagocytic inflammatory cells that traffic to the site of the infarct, which you can see here in false color. So you see the cells coming and also leaving. Excuse me. And to give you an idea, this whole imaging session took under 13 minutes to image the mouse heart. And you can imagine the mouse heart is very challenging to image because it is very small, and the heart rate is also very high in mice compared to humans. We've also looked at non-cell-based images. So this is looking at bone matrix injected in a defect in the knee joint of a mouse. And then here, in this case, we followed it for 8 weeks. So you can see where the knee joint is with the yellow arrow, and that's where the nanoparticles were injected along with a bone filler. And we've looked at various different cell types as well. And again, in these images, the cells are shown in false color over the grayscale anatomic MR scan. We've looked at primary human dendritic cell subsets because that was our interest for a long time, but also different types of stem cells and inflammatory cells. You can also take this ex vivo and make 3D cell phantoms to look at cell migration, for example, in a longitudinal manner, where you can then quantify the cells migrating in 3 dimensions. And this can then be corroborated with what we see in the patients as well. So let's talk a little bit about multimodal imaging, because the nanoparticles that we work with, they work not only for fluorine MR, but also with photoacoustics and fluorescence. So here we have the same cells in the same mouse that was injected with 3 modalities, with either 100,000 labeled cells or 1 million labeled cells. With photoacoustics, you see the cells in green, and then you also get signal from deoxy and oxyhemoglobin, which you see in blue and red. And the same mouse with fluorescence and the MR, which you're now familiar with the fluorine in false color. We can also preclinically adapt the particles for nuclear imaging. So if you add a radionuclide, you can see them in SPECT and PET. And we also see them in ultrasound, as you see here, injected in the lymph node, which previously was dark and then becomes bright with the injection of the nanoparticles. The ultrasound contrast was really puzzling because typical ultrasound contrast agents, you may know, are very large gas-filled microbubbles that oscillate under exposure to ultrasound, and that's what you're looking at here. Whereas our agents are very small, they're only 200 nanometers as opposed to 3,000 or 5,000 nanometers in diameter, and they don't contain a gas. So this was quite puzzling to us as to why we see these agents under ultrasound. So to try and understand what was going on, we carried out a whole bunch of structural studies on the nanoparticles. And I won't go into too much detail because I think that's less interesting to this audience. But what we found was that the structure of these particles is a very unusual multi-core fractal structure. So it's not a typical core shell structure, but instead we have these multiple smaller domains of perfluorocarbon surrounded by the PLGA coating. And this has very, not only was it a unique structure, it has very interesting in vivo properties, because when it degrades to these smaller domains which remain suspended. So they remain stable in aqueous environments. And we've shown this using atomic force microscopy or dynamic light scattering. But what happens in vivo is that when you have this multi-core structure, compared to a typical emulsion or even a capsule, which is exactly the same PLGA and trapping the perfluorocarbon, but with a core shell structure, what we see is that the clearance is very different. So here you're looking at the liver and the spleen of mice that were injected intravenously with the same amounts of each of these labels, but the clearance of the multi-core structure is almost 15 times faster. And this is really important because it is known from the past that perfluorocarbons can have very long retention times in vivo, in the order of 250 days in mice. And this is very difficult because what that means is if you're carrying out cell tracking, you have no way of telling whether you're looking at your live cells or simply the label that has not been cleared. So fast clearance is really important in order to carry out cell tracking. So we've labeled many different cell types of these agents, many different primary human dendritic cell subsets, but also natural killer cells, Carti cells, and so on, and also different kinds of stem cells. Of note, the label uptake varies per cell type, as you would expect, because some cell types are larger and more phagocytic than others, and this directly impacts the sensitivity of detection. It is vital to show that the labeling or any other kind of modification you're carrying out does not affect the cell's functionality or significantly affect their viability. And after several discussions with both the EMA as well as the local regulatory bodies, we have shown that labeling does not affect cells when compared to non-labeled cells. And this requirement, of course, depends on your particular cell type. It's also important for us to show from a regulatory perspective that the label is contained inside the cell and not outside the cell. So we're not injecting free label to the patient, because that leads to a different regulatory pathway than if you're injecting label cells. So we've shown this using confocal microscopy in various cell types where you can show that the fluorescent label is inside the cell. So in order to get regulatory approval, we need to show in vivo data, but this is really built on a huge volume of in vitro data, where we really need to show that there is no toxicity, both of the labeled cells and of free label. And also we need to look at the fate of the label as well as the fate of labeled cells, both in vitro and in vivo. I'll just show a little bit with the nuclear medicine-based cell tracking that we have done, both using SPECT and PET. So it's the same nanoparticles, which are produced in the same manner, except that they're labeled on site with either a tracer for PET or a tracer for SPECT. And both of these are clinical imaging modalities. So here's an example of THP1 cells, which is a T cell type homing to a site of infection, which you see very clearly here. This is a site of infected muscle, and then you can see about, in this case, about 27,000 cells homing there after intravenous injection. And we can also do this with PET, where the cells traffic to the tumor. So we've shown this both in an infection model and in a cancer cell model. So just very quickly to also move on to non-animal-based applications, we've also been looking at drug delivery in plants, because there is a lot of need in crop plants such as grapevines, but also other crop types, to deliver drugs to form depots in the stem rather than spraying drugs onto the leaves of plants, which is currently what is often done, because the infection is often in the stem rather than in the leaves. So what we did here was we injected the nanoparticles, which can be loaded with a therapeutic agent, into the stem of grapevines, and then we looked at the formation of a depot over several days. And you can see that the nanoparticles stayed at the site of injection, which is what we want to allow for drug release at the site. So I hope I've given you a quick overview of the various imaging modalities that can be used for preclinical and eventually for clinical use as well, and some with the same imaging agent. So when someone talks about the work that has happened in the past, it always sounds really easy, but in fact, there have been a lot of hurdles towards clinical translation. If you are working with nanotechnology, you probably already know that it falls into a regulatory gray zone, and it is often very unclear for regulatory agencies how to handle nanotechnology, and this makes it very challenging and very expensive for SMEs, both to get private funding and to actually reach the clinic. In our case, because we're also working with an imaging agent, this led to even more complications, because imaging agents are often classified as drugs, even though they are not pharmaceutically active. And if you look at the market today, there are no multimodal imaging agents available, and very few agents, or actually no agents specifically approved for cell tracking as well, simply because the regulatory hurdles can be very high. In addition, when we are developing an imaging agent, it is not only the need for the imaging agent that we need to look at, but from a business perspective, we need to look at the availability of scanners for these imaging. Now, most of the centers that do cell therapy trials tend to be in well-equipped research hospitals, so they have imaging scanners like MRI or PET or SPECT. But if you go to smaller hospitals, this is not the case anymore. So then it makes much more sense to move to a modality like ultrasound, which is much more widely available and much cheaper than something like fluorine MRI. But then you lose the advantage of quantifiability of cell numbers. So these are factors that you also need to take into account when building your business plan and setting up your SME. In addition, when you're working with cell therapies, we need to have the infrastructure to label the cells and then to characterize the cells. And this either needs to be done at the site of injection to the patient, or if there's a central processing facility at that point, but then you need to look at the logistics of transfer of labeled cells from the central facility to the site where the patient actually is. So all of these factors, which are in reality quite far from the science of developing the imaging agent, are also really important and need to be considered when building your business plan. And in my case, as Pascal mentioned, we were part of the Venture Challenge, which allowed us to get, because as scientists, of course, we are not trained in building business plans. That's not what we are taught to do. So part of the Venture Challenge or other courses like that, they're really useful because it teaches you all of these other aspects that you need to take into account, because simply having a good idea doesn't mean you have a good product. Because something is exciting academically doesn't mean that it's going to succeed in a commercial world, unfortunately, but that's how it is. And of course, we need to really look into quality control and so on. So we've been very lucky that a lot of our funding has come from the European Union, which enabled us to do the translation to upscaled GMP production. Because if you have worked with GMP production, you know how much of an administrative nightmare this can be, to put it mildly. So it is really it's really useful that the European Union has now recognized that translation of nanotechnology is extremely challenging and is working in many different ways to help support SMEs working in the nanotech field. So I hope I've given you a short overview of why it is important to track therapeutic cells and how imaging can help. That this information is needed for efficacy, for safety, for regulatory compliance, and also for fundamental understanding. It is unclear if imaging will be required by regulatory bodies for cell therapies in order to support reimbursements. But if that happens, of course, that means the market will be much bigger for this kind of product. But overall, there is a strong unmet need to quantify therapeutic cells in order to optimize them in clinical trials, but also in the clinic at a later stage. So with that, I would like to thank all of you for your attention, and again, the organizers for the invitation. And I'm happy to take any questions.
Thank you very much, Professor Mangala Srinivas, for this excellent talk and presentation. Unfortunately, we don't have the applause that we usually can have in a conference room. But definitely you have convinced us of the usefulness of imaging technique to improve cell therapy. I would like to start with the first question, if you mind. So how do you deal with the market demand? So how have you been able to measure the demand on the market for such technology, and how do you deal with this in your business approach?
Yeah, so there are a couple of ways to do this. One is a theoretical approach to try and calculate how many clinical trials are going on with cell therapies, and then estimate how much of that will be a market for us. That's one way to do it. The other way is, because my SME is linked to an academic group, we can also see, for example, how many people approach us for, can you send us some nanoparticles to try, right? But of course, does that translate then to somebody buying the nanoparticles is always up in the air. But one of the challenges, I think, for us is that there is no competitors that we can look at to see what their market is. And in a way, this is a new market that we are building because cell therapies also are new. So it is all a little bit of the Wild West here, but okay. So far, so good.
Okay, thank you. I see a question from Fatin Dachinar. What kind of challenges, especially in terms of legislation, have you been facing for the study, implementation, and dissemination of such cell therapies?
What do you mean by legislation? Do you mean regulatory? Or
I guess, yes, the regulatory aspects and probably regulatory aspects in different countries might be different as well. So yeah.
So actually, if you look at the market for imaging agents, there hasn't been anything new for about 10 years or 20 years. And this is because for a long time, imaging agents have been classified as drugs. And once something is classified as a drug, it becomes very expensive to develop. And then it's not worthwhile for a large company to invest in. When it comes to specifically agents for cell therapies, and I alluded to this in the talk, in my talk, the reason we're going with labeled cells as opposed to injection of free label is because they fall into different regulatory categories. So if we are injecting a labeled cell, this means that the active product or the therapeutic product is the labeled cell, not the imaging agent. But if we inject the imaging agent, then the active product is the imaging agent. So it's a little bit of a small trick, but this allows us to bypass the regulatory requirements for an intravenous drug and rather go with the labeled cell. And that greatly reduces the cost, because then we only need to show ex vivo that the labeled cell functions like a non-labeled cell. And this you can do with standard assays like T-cell activation or migration or whatever your cell type is. And that makes it a lot simpler. And we see that both with the EMA and the FDA.
Okay, thank you. It's very interesting. Another question from Beisanur Chaka. Thanks for the presentation. It was very exciting. I have a question. Is there an assessment of the half-life of the injected nanoparticles?
Yes, we need to do that in mice. We cannot really do it with labeled cells because it depends on the cell type and the lifetime of the cell. But we have done it by injecting free label into the mice to see how long it survives. We were also quite lucky that we were funded by the European NanoMedTech. I forget the name, but they had this project for a while where if you are selected, you send them your nanoparticles or some kind of nanotech, and then they do a whole array of in vivo testing to look at toxicity and so on, all the standard tests that would be required for a regulatory filing. And I think those kind of initiatives are still going on, and that's really useful for small companies like ours.
Mm-hmm. Okay. I see another question yet from Professor Rana Sanyal. Amazing talk, Mangala. Thank you very much. Could you elaborate a bit more on the regulatory aspect of adding a diagnostic agent on the therapeutic? Did you have to do all toxicology studies separately for the diagnostic again?
So when you're talking about a diagnostic agent, then you're talking about an application rather than adding a drug. So the imaging agent itself is not a therapeutic. It's simply classified as a novel excipient. If we add a drug to it, then it becomes a therapeutic. So and then a diagnostic is another layer where you're looking at the clinical utility of something. So these are both very different. Once you add a drug to the imaging agent, you do need to repeat the tests. It might be slightly less than you think, because probably the drug you're using already has a lot of toxicity data on its own, but it will need to be done again for every modification that is made to a nanoparticle. And that makes it, of course, much more expensive. If you are then looking at a diagnostic application, that requires a large randomized clinical trial to really show efficacy as a diagnostic. And that's, again, a different layer.
Okay. Thank you. I will take another, if you don't mind, we will take another question. Which viral vectors do you think will be more successful for Carti cell therapies? This is from Rumeisa Ekishi.
Honestly, I have no idea. It's quite far from what I do.
Okay.
I think that's for the Carti cell developers. We have worked with a few in the past, including some large companies. And there is a lot of need, I think, for cell tracking in the CAR T-cell field, but I have no idea which vectors would be better.
Okay. Okay, there is another question from Professor Cengiz Öztürk.
I think I'm entitled to ask it like here on screen. I guess some of us are on screen, and I couldn't type it on the Q&A. I must be on panelist. So thank you. This was a great talk. It was very exciting to see all the preclinical data and eventually a pathways for clinical utilization. So I was wondering, so there is clearly an unmet need on human cell tracking. Mm-hmm. And you of course, there's a huge data that you presented and research going on the preclinical, multimodality agents and trackings and for this work. But eventually, what will be translate into human imaging? Do you think the PFCs will be the final agents, or more PET agents or SPECT agents will be utilized for that? That was not clear to me. All this preclinical work is necessary for the basic science and all the necessary background, but what will be eventually a GMP agent that will have a success and also widespread utilization in routine clinical practice that cell therapy promises to do in many different things?
That's a great question. So actually what is approved for the clinical trials that we have at the moment are the fluorine-based cell tracking, so not the radioactive version. And one of the strengths of fluorine-based cell tracking is that there is no radioactivity involved, which makes it a lot easier for the patient. And also, we're not restricted by the half-life decay of the radioactive isotope. So even with zirconium, you're limited to maybe a week of imaging time, whereas with fluorine, you can image for potentially for 8 weeks or months, as long as your signal is still there. But your question was different because even if you show success with fluorine imaging, there is still the difficulty in that it is a relatively new imaging modality and it is not present in all the clinics. A lot of the bigger hospitals have MRI scanners and many of them can be adapted to fluorine MR, but it is still an expensive, rather specialized technique. PET and SPECT are much more widely available. In addition, they have higher sensitivity than fluorine MR. So for us, from a business perspective, it made sense to kind of divide our risk and to look at both approaches because it's unclear which one will be more applicable. But the trials that we have approved are all with fluorine MR. We would like to really look at ultrasound and photoacoustics as well, because these are relatively cheap imaging modalities, ultrasound especially. But the information that you get is much more limited than with MR or with PET or SPECT.
Okay, thank you, Mangala. I have one last question or comment from a previous person who asked a question, Professor Rana Sanyal. Professor Sanyal, would you like to ask your question directly on live? I think it might be easier if you would open your microphone.
Maybe they can't, Pascal. I am not sure they can make her panelist. Maybe attendees can only ask questions.
Oh, okay, so they cannot.
In written form.
All right, so then I will read the question again. Rana said, I meant the cell therapy as the therapeutic, not as diagnostic application, but only for tracking your cells therapy in humans.
Oh, I see. Yes. Yeah, so the question, I guess, that you're asking is, will the agent, the imaging agent, need to be reapproved for every different cell type? Is that what you're asking?
So I'm waiting. We leave her a couple of seconds to answer on the chat. Rana is using the chat.
If that is the case, then it is not, I don't think that will be necessary, because from what we understood from the EMA and also preliminary discussions with the FDA, is that if we are classified as an excipient mix, then it's independent of cell type. And that is really, so let me first finish this line. So it's really important for us, and I think for any developer like this, to not be linked to a specific cell therapy or a specific single application, because that's very restrictive for your product. So for us, what we are going for is a generic cell label. And from what we've understood, as long as we stick with the ex vivo cell labeling approach, it doesn't matter what cell type you have. Now, if we add a radioactive agent to it, we are indeed modifying the particle, because now you have the the finished particle, which is more or less the same, but you have the radioactive isotope. So we do need to do some toxicity again. I don't think we will need to do all of it again because the radioactive isotopes are pretty standard in the clinic already. And then the particle is also we already have all of the data with the particle. So it is a relatively small modification. What we do need to look at then is that we don't overload the cells with radioactivity, because this is a slightly different approach than what is typically done with PET or SPECT. So you have a lot of cells that might congregate in a small area. So you might have suddenly a lot of radioactivity locally. And that is something that we need to control for.
Okay, thank you. Thank you, Mangala. So at this point, I don't see any additional questions. So I think it is time for closing the webinar. Thank you very, very much, Professor Srinivas, for this excellent presentation. You have been the first keynote speakers of the webinar series, and it has been a very exciting moment today.
Thank you for the invitation.
You're welcome. Thank you, Professor Öztürk, for the presentation of the center. I would like to thank all the participants today who have joined this first webinar of the series. The next webinar will happen in about 2 months, in June 2022. Stay tuned. We will make further announcements. And with these last words, I would like to close the webinar. Thank you and goodbye.
Thank you. Bye.
Thank you.